Welcome to the Neural Implant Podcast, where we talk with the people behind the current events and breakthroughs in brain implants in an understandable way, helping bring together various fields involved in neuroprosthetics. Here is your host, Laden Yurachek.
Hello, everyone. Welcome to the Neural Implant Podcast. Today is a repeat guest, which we don't do often. It's Dr. Kip Ludwig, and he was one of the first people on the show, I think like episode 2. But yeah, pretty much Kip taught me about bioelectronic medicine. I'm just like, what? That exists? And it was, I remember that. It was pretty formative time. And at the time you were at Mayo Clinic, which you are no longer at. You are now at University of Wisconsin. I wanted to have you on because I wanted to refresh some of the stuff that you had talked about, and then also talk about some new stuff. And honestly, you didn't get as much exposure as you should have in the beginning episodes. And so I want to give you the exposure that you deserve and not the 40 or 50 downloads that you had originally gotten. So Kip, pleasure to have you on the show. Welcome.
I disagree that I deserve exposure, but thank you very much.
So do you want to introduce yourself, maybe refresh what you have been up to historically, and then what's been going on in the last 4 years as well?
Yeah. Hi, everyone. I'm Kip Ludwig. I actually started out at the University of Michigan in brain-machine interfaces, and then went to a company called CVRX, where I developed their minimally invasive device to stimulate the carotid baroreflex for hypertension and heart failure. I then went to the National Institutes of Health, where I led a lot of the translational programs for neurotechnology to try to get academic research and small business research created a lot of the public-private partnerships that were part of the BRAIN Initiative, launched the S.P.A.R.C program before Gene Civilico came and took over it. Then I was at the Mayo to run my own lab in neural interfaces, and now I'm at Wisconsin, and I'm the co-director of the Wisconsin Institutes for Translational Neural Engineering, otherwise known as Whitney.
So you've seen all parts of this. You've kind of the triumvirate. You've collected all the Dragon Balls and are now able to have a very good point of view of the field because you've had government, academia, and industry experience. So yeah, I guess at a high level, what's your impression of the field or what's it like?
Yeah, I think the first thing to ask me is what the hell was I thinking? But no, it was really interesting. I actually went to industry quite deliberately because I started off in brain-machine interfaces. And this is right about the time when the companies associated with implantable brain-machine interfaces went bankrupt. And so I looked at it and I was looking at a postdoc in Yuri Basaki's lab and some other labs and decided that, okay, if these things that are showing incredible proof of concept in humans don't really have a viable business case, I want to go out to industry and understand, how do I create something that can get to people and sustainably help? And I'd like to say right before I did my dissertation, but honestly, it may have motivated me to do my dissertation. Eric Lovett at CVRX reached out to me based off of my LinkedIn profile and had me in and offered me a job. And then I got to go to CVRX. It made me finish my academic research very quickly. Somehow getting paid $44,000 as a postdoc did not match to getting paid $96K starting as well. So there was a little bit of math there. And then I spent the first 6 months regurgitating every idea I heard in academia. We were going to add these drug coatings to grow nerves towards things and all sorts of things. Really looked stupid in every meeting, probably for about 6 months. Kept on getting shot down by the way they do it in business, which is they work back from reimbursement. They think about supply chain. They think about a lot of the things were overly complicated, which means it's going to fail a lot more chronically and things like that. And say, okay, I learned a lot about developing things that are tractable that can actually get to humans. And that's really changed my perspective. Then I went to the NIH and I saw a lot of academic research sitting on a shelf or even getting to cool proof of concept in humans, but having no path to creating a business, and did what I could to build programs to help better build and translate things that we might have a sustainable market, and did what I could. And then went to academia because there were some things I couldn't address and still didn't do for me what coming up with a new idea, testing it and seeing it work in a lab does. I've never stayed up till 4 because I came up with a new program idea when I was at the NIH reading papers and trying to get it done, whereas I do that all the time for scientific ideas.
And that's why I like talking to you, because you have a unique perspective on this and a little bit, I don't know, pessimistic or realist or something like this. And I really appreciate that. It's actually really refreshing. So before we started recording, Yeah, but for me, I especially like it because it's very German, and I really like Germany and everything like this. So yeah, exactly. That's where that comes from. But before we started recording, you were talking about drug-resistant drug coatings on neural devices.
Yeah.
Why might not that be a good idea?
Yeah, and it can be a good idea. It's just a lot of complication. So I've dealt a lot with supply chains. I do a lot of advising on developing devices through regulatory testing and things like that. And now you have to deal with dosing a drug and developing a device. It's very different supply chains. When you go through regulatory approval, now you have to deal potentially with CDER, which is the drug division of the FDA, CDRH, which is the device division, and going through the Office of Combination Products. And as I mentioned, the more government bureaucracies that you have look at things, it becomes a little bit more scary and daunting, but they're also not individually as proactively helpful. Often they start going much more by the book because there's another division of the FDA that's also involved. And so they feel a little bit less comfortable themselves. So I had mentioned that there's multiple companies like Medtronic that have severely downregulated or scrapped their, say, drug delivery devices portfolios and things like that, especially for brain delivery because of this complication. It just becomes harder to make a business.
Yeah, definitely. And that's something that a researcher might not necessarily think about, but somebody who has that experience and potentially has gone through that purgatory of at the same time burning money and waiting for a few government bureaucracies to work themselves out. That's something I guess you don't forget.
It also depends on what it's for, right? If I'm doing it to another 2 years of recordings for a brain machine interface that's already a limited market, then those increases in complexity and cost and things like that are really problematic. But if you're doing it for something else with a larger market where both things are really critical for the core function, then it still might be worth doing.
Yeah. So essentially, unless you're getting like a 10x benefit, then it's almost not worth it. If it's just like a 20% improvement, it's not worth the hurdles.
Yeah. Multi-channel DBS is the same thing right now, where Medtronic shelved Sapiens after a $200 million acquisition. And part of it was they didn't know whether 40 channels was going to grow the market for them. In fact, right now, we still don't know if it's going to maybe help with everyone's slice of the pie of an existing market. It doesn't seem like it's going to get over the hurdle of distribution, referrals, and people who are just generally afraid of having a burr hole put in their head and a 1.2 millimeter lead put into subthalamic.
Yeah, for sure. That is something that, I don't know, I've been salty about this Sapiens acquisition and then the shelving, but it's different calculus on either sides of the transaction. So you got to really take that into account. But the point of this episode is to go through some of these theories and these ideas that you've been having and I've been hearing. So last time we talked too off the air, you had been talking about over time, studies have not really shown better efficacy. And I think it had to do with the number of channels. Was that correct?
Yeah. So I think what you're referring to is Warren Grill did a nice paper, I think somewhere out in 2013, where he was looking at spinal cord stimulation for pain. And he essentially mapped out over time from, say the late 1990s until now, or at least until 2013, all the quote unquote RCT studies that were done. And there were over time, there was like 100 of them. He was actually drawing from like 3 critical reviews. And if you look at any one of them, it would say there's a 90% responder rate to SCS. There's a 10% responder rate to SES. And if you put them all together and map them over time and you draw a line, it's been 50% responder rate, regardless to some extent of modality, which is why I never pay attention to any one or put too much emphasis on any one clinical trial. A lot of times with devices, we're dealing with ends of 30s or 60s in terms of subjects, when in drugs, they'd be doing 10,000 subjects. And so until you see how it bears out over time, like Nevro had these fantastic results for high frequency therapy that were 90% or so responder rates that in subsequent studies have not been nearly 90%. And then talking to people at Nevro, they'll tell you that we were very careful about things like our exclusion criteria and things like that, that would not be clinical deployment, because it's interesting when you're doing those trials. We had these conversations at CVRX all the time, where you want to exclude people who would be, less likely to be responders or tougher conditions and things like that to actually pass your clinical trial. But when you still want to have them included as part of the people you can market to, ideally once you're FDA approved or once you're CE marked. So there's that kind of fine line. And often then the post-clinical trial results aren't quite as compelling because they're broadening the people who are being included. Conversely, though, there's also this learning effect over time where sometimes it gets better just because after the clinical trial, that's often the first time you're deploying it in 40 centers with 250 patients, and you learn a lot from the process, and you get a lot of information, and you get better over time as well.
That is interesting, and it's disheartening that it's not getting better.
Any of this stuff works. You can go down the list. Deep brain stimulation. It started off being, okay, lesions of subthalamic nucleus stop some of the symptoms. Let's do what we think is an electrical lesion. And then it became, okay, we're doing a functional information lesion where we're stimulating 130 hertz where no normal physiological information can get through. Then it's evolved to things like maybe antidromic activation of motor cortex, or it could be stimulating glial cells to release neurotransmitters that cause inhibition. It could be plasticity mediated effects over time. Same thing for spinal cord stimulation. There's roughly 7 or so major or more theories about how it even works. And probably the same thing with vagus nerve stimulation. You can go down this list. We actually don't definitively know what we're stimulating for effect in most of our largest markets for neuromodulation therapy. And we don't know what we're stimulating for side effect in many cases. So how do you make it better? That's why I went to the NIH. We needed new tools to be able to visualize on and off target engagement. We needed to understand our animal models. We don't even understand how predictive our animal models are of the human condition. So I wanted to create systems where we do parallel translation. We're looking at the variability in the human anatomy. We've never done a study with DBS in animals that also includes L-DOPA, which is these patients are given L-DOPA to ameliorate their symptoms. They adapt to it over time. It changes the neural physiology. They wind up getting L-DOPA-induced dyskinesias. But our animal studies do none of them for any of these therapies.
Interesting. I didn't know that. Yeah, I do want to get to the animal studies later, but yeah, no, I think that this is absolutely true. Like we have no idea what's going on. And Elon Musk has said, it's like kicking the TV. Like it's not working, it's flickering. And then the old CRT, you kick it. What did that do? Like, we don't know, but it works now. So that's very much what it reminds me of. Yeah, it's just like resetting the system.
Yeah, you unplug your computer, you turn it back on. You don't know exactly what caused it to crash, but hey, it worked. But predicting that when it will work even, you can't do it until you figure out the mechanism.
Yeah, definitely. So I want to talk about the animal models. This is something that your group has been working on, and I found very interesting. Everybody uses mice, everybody uses rats, rat models, but that might not be very accurate because humans and rats are
different sizes,
different smelling abilities, like all this kind of stuff. But yeah, different physiology, right?
Yeah, in many respects, yeah. For spinal cord stimulation, if you go to a pig or a human, 90% of the current is estimated to shunt in the CSF because you got a lot more CSF between your electrodes and the cord than in mice or rats. So in many cases, we're completely misunderstanding the transform between what's activated in a rat and what would be activated as a human. But the bigger problem, I think, is almost every one of these therapies is limited by a side effect. And the side effects we tend to ignore, and they tend to be simple things like off-target muscle activation, perception of pain, perception of paresthesia. And those things depend on how close the local anatomy is, scale. We tend to almost rightfully ignore them in a rat because, well, you're stimulating things in a very small area. You get off-target stuff that you won't get, but that doesn't mean you don't have to figure that out. Almost every neuromod therapy, you go through all your electrodes, you titrate up the side effect, you turn it down a notch because you can't go to the side effect. And then you try to figure out, okay, can I still get the effect I intended at one of these contact points? I programmed a lot of human patients and it's maddening. So for CVRX, we were looking for blood pressure drops and for hypertension and heart failure. And the off-target effect would be neck muscle activation, really low threshold fibers that are easy to activate. And you would just start to see the heart rate going down. You'd see it go 5 beats per minute. You see the pressure go down 5 millimeters of mercury, and then you would get the side effect. And you're like, but I just got to the cusp of the dose response curve. And you're often, because these are invasive devices, dealing with patients who have very severe problems. Your chances of dying in the next 5 years if you have heart failure are very high, and drug therapies don't do much. If you have intractable hypertension, not responsive to drugs, and you have a blood pressure of 260 over 160, which was patients we'd see in clinic, that's not a good long-term prognosis. So to see this be limited by side effects is just maddening. But we never developed devices to understand the side effects and avoid them. We didn't try to figure out what's the human anatomy for all this. Right now, they still don't know what human anatomy creates the side effects for spinal cord stimulation. It's been around for 20 years. Vagus nerve stimulation, it's been around for 20 years. We just published a paper. These are the likely pathways that are causing you the side effects that you should design your.
So what's the solution? Use more human testing? Or, I mean, that's not what you were suggesting, was it?
No, it's really create systems where we do human cadaver work to understand the variability of the anatomy. We do small animal work, often with optical tools, so we can directly visualize what's being activated for effect or side effect. We do large animal studies that kind of bridge the gap. And then we work with great people like Scott Lemke and Warren Grill and Luis Lujan at Mayo to do computational models where we can start bridging the scale and understand if the pig has 40 fascicles instead of 6, what does that mean? How does that change neural activation for the on and off target pathways? I think that's the other problem we have with animal models. They tend to have inherently less variability because we're getting them from the same providers with the same genetic lines in many cases. If you look at the human vagus, it can have cross connections to the sympathetic trunk, the carotid sinus nerve, that are not nearly as variable.
Yeah, definitely. So you were suggesting more pig models, right? Because that's more accurate or more similar to human models.
Oh, well, it depends on what it's for. For vagus nerve stimulation, like the dogs and NHPs only have a couple of fascicles, really. Whereas the human, we have some micro CT studies now where they might have 9 fascicles at one point on the vagus, and then half, a centimeter later, they might have 1. So it's just a different beast, both literally and figuratively.
It kind of puts all those things into question, I guess. And I like questioning basic assumptions. But yeah, what else is there that most people would disagree with you on?
Well, that's an interesting question, the way you worded it. I'm a Buffalo Bills fan. Most people aren't. It's funny. I get a lot of positive feedback for the things that I say. And I think it's because it's just acknowledging a reality as opposed to ignoring it. I've never really had anybody come back and say that's wrong with that. It's obvious to some extent. I'm not that smart. Most of what I say is just obvious. It's just I'm willing to say it. Hey, you're trying that in an animal model and you're trying it acutely, and you're going to try it in chronic human beings who've been dosed with these drugs for 20 years that are a completely different scale, that have completely different fascicular organization. Maybe you should be thinking about how that might be different. And honestly, I'm just preaching from my own experience. At CVRX, I had gone in and designed a really good dog stimulator that wasn't as optimized as I would have liked for the human condition, because we weren't really thinking through what's the off-target effect substrates, how do we design around it, what are the differences that might be very important. But that's the problem with a small company. You get venture capital funding, and then you're on a short leash. You got to do things in a short period of time. So you take your shot at goal, you adjust it where you can, but you don't have a lot of time to do science or learn from the process and substantiate it.
Yeah, the VCs don't care about science. They want a return on investment, right?
I mean, yeah, the people who are coming in at pre-seed want to get, or seed want to get paid at Series A. So they're hoping, they don't even care if they do. I'm sure they care if you get the market. I don't want to be totally nihilistic about this, but they want to get paid at your Series A or your Series B. That's what other people don't realize. They don't realize that your early VC is not coming to make money 15 years when you get market approval and reimbursement and stuff like that. They're getting paid at your next funding event.
Yeah, definitely. So what is, short of spending a few decades following in your footsteps, like what's the solution? How do people gain this perspective? Maybe they should just have you on speed dial, or what?
I do have a lot of people call me when they're first doing things and talk about what they might run into. I would say most of the things you've heard about or are popular right now, I've probably talked to people involved at some point, and that's just common. And there's people who do that routinely, whether it is a Tim Dennison, former VP at Medtronic, or a Hunter Peckham, or a Bob Kirsch, or a Kevin Kilgore, who have done this for FES for forever, or a Dustin Tyler, or a Florian Sulzbacher. There's all these people who have done it who really are going out of their way to create conference sessions and whatever that are different than the hypey ones, and just say, no, this is how you do it. And this is what you have to know. So to some extent, you just have to be willing to go to those things and realize that it's really cool to get proof of concept in nature. And that roughly means nothing for helping a human being. The 90% of the effort is in that last 10% to take something from proof of concept in animal models and get it to something that can reliably help humans improve it. and then have it be scaled so that other people can do it. It's a crazy thing, actually, when you go from doing an animal study to large-scale human studies like I did, and seeing essentially you're trying to teach 40 separate centers to do the experiment, input their own thoughts on it. They deviate from what you tell them. You can't, when it gets to that scale, you can't keep it from happening. So the best advice that I got was from, again, my former boss, Eric Lovett. He's like, look, you need to make the Neo device, which is the one that got recently PMA approved by CVRx, so good that it will work no matter what stupidity is thrown at it when it's scaled out.
Yeah, idiot proof it pretty much. And granted, it is more educated, more trained people using it, but still, like you have to be able to do that. So it sounds like it's more of a mindset, like more versus, hey, go do this and then do that. So it's more of a mindset like, hey, just realize that shit happens essentially.
Yeah. Well, and go into it knowing when you first go to humans, the jump is so large from animals to humans. I've talked to people who, there are, again, nihilistic people I don't agree with who think animal data is roughly useless because it's limited predictivity. I think I like to create systems where we can make it more predictive, but when you jump to humans, you're going to learn a lot and you're going to have to change some things. So don't go into it thinking that the device that you developed in animal models will work really well in humans. You're going to learn a lot and it's an opportunity to get better and make it better. But then you got to figure out how to fund it.
It reminds me of the joke. We figured out how to cure cancer in rats like 1,000 times, but not in humans.
Well, no. So I got to shepherd the spinal cord injury replication contracts for a while when I was at the NIH. And these were essentially 10 seminal studies that started in rodents and then eventually went to humans and failed. And what they wanted to do was figure, was the rodent model just not predictive? Could those rodent experiments even be replicated by an outside lab? So was there a problem at the rodent level? Do we need to be doing different models? Do we need to be understanding, not wasting our time? And roughly only about 1.5 of the studies actually even replicated from rodents to rodents. And one of them, so we started it out and you couldn't talk to the other investigator when you replicated. You had to go just by their methods and that just didn't work, but we didn't want them to be biased. Then we had them talk to the other investigator. And in one particular case, that half replication was because they used an animal, a rodent line from a different provider. When they got the same rodent, then the therapy worked in rodents. And of course, if that's the case, if I have to get the same genetic line, you're done in humans. To heck with even the scaling to humans and things like that. It's just, if it only works in a very specified genetic rodent model and not others for spinal cord injury, the likelihood of helping humans is nil.
Yeah, really. You're not making a good case for this animal studies. It sounds like you should go direct to humans as soon as possible.
I don't agree with that either. And that's because the amount of information you can get in humans is much less. I do think we need to focus a lot more on, you're not going to figure out target engagement. The reason we don't know target engagement in all of these therapies, in many cases, is because we're sticking them in humans. We have no way of measuring what we're activating for effect or side effect reliably in the home under use situations. So what you need to do is do animal studies and human studies together and use them to complement each other. Use the techniques that you can in animals that you can't use in humans because they'd be too invasive or involve genetic modification. But understand that what you just showed in a rat, you need to be very cognizant of what it's going to be like in humans and how it's different. And that we're not so great at. We like to say we just cured cancer in a rat. We like to say we just cured cancer based off of rat data. Done a great job of treating spinal cord injury in a rat, but we still have some distance to go in humans.
Yeah, for sure. I don't know. I was actually thinking that human studies would be able to give more information because you could say, what are you feeling? Or I'm feeling a hoarseness in my throat or something for vagus nerve. And you wouldn't be able to necessarily tease that information out of an animal that can't speak. And you have to think of some like convoluted experiment to be able to get that information out.
You are dead right. And that's important. That's why human studies need to be done in concert to some extent. I'm a big fan of taking advantage of existing surgical opportunities to get some information in humans as well, so that they're not put at risk for what's completely exploratory at that point. But right now, if you want to figure to get that hoarseness, am I activating A alphas going to the recurrent laryngeal, the superior laryngeal pathway? If I need to figure that out, I need to do an animal model. I need to do detailed recordings and I need to do transections. And in many cases, a lot of our human stuff is very contaminable as well. There's been a lot of studies. There's been a recent study for nerve stimulation where they're stimulating the ear, where they found out a large component of the somatosensory evoked potential goes away when you do a muscle block, because it's not afferent activation of the brain. It's actually just muscle bleed through from, you got a lot of muscles you can activate. And that's been the case across the board. We thought there was a possibility that human VNS was working through C fiber activation, and that there was a clinical study that suggested that you could essentially C fiber activation at the lowest settings on the IPG. And the reason being, and we showed it in our recent paper in pigs, they were mistaking a muscle activation artifact for the conduction velocity delay of a C fiber, and was actually something created by the long pathway through the recurrent laryngeal. So without confirmations, it's really easy to get tricked in this. That's why we've been wrong about mechanisms.
Wow, because you're dealing with the body and the nervous system with so many extra feedback loops or extra side loops. It can just loop back on itself and give you this crazy data. Wow, that's crazy.
Don't ever date electrical stimulation. It will cheat on you. It's very promiscuous. It also creates a lot of effects in non-neuronal cells that may be important. It's also, people think they're stimulating wires. We activate those wires in both directions when we activate them in a way that does not happen physiologically. So electrical stimulation is very dirty. There's no way around it.
As humans, you and I, are we biting off more than we can chew? Is this something that we need to wait for AI to be able to solve?
You can't do that, right? Because there's people in need that aren't being treated by drugs. And so you do what you can. I understand the phenomenology. I understand why they do it, because you have to. And if you take some of the stuff that we've seen across the field, if you take a severe hypertensive and you can turn on the device and off the device and have them go from 260 over 160 to 80 over 40, which we saw in maybe 15% of the patients or something like that. Could save their life. So you have to do it. But we have to understand how little we know. And just because what we always assume is, a friend of mine at Mayo used to call it, we have a lot of bioplausible bullshit where we say we're going to get this effect through this neural pathway. We never prove it. We just get the effect. So we assume we're right. And the history of neuromod is we've been wrong more times than not when we made that, even if we get the effect we want.
But is that bad? So for me, I'm more engineering minded, more practical. It's just, if it works, right? And why question it? When the Wright brothers made the plane, they actually didn't know the theory of flight. They didn't know what was actually causing flight. And that would only come later.
Yes, when they understood the theory of flight. So yeah. I think for me, it's great when there's phenomenology. When there's something that says, hey, we can actually get an effect that's helpful. That's a great reason to go back to animal experiments or do parallel studies in humans and animals to figure out mechanism to optimize it. But I've listened to your podcast enough to know that many of the people are lamenting DBS electrodes haven't changed in 20, 30 years. None of the neural implants have changed in 20, 30 years. Well, part of the reason is we don't understand the physiology of how we're interfacing enough to make it better in a clearly useful way that has a market. We don't know if 1,000 electrodes helps you for brain-machine interfaces or for DBS or not. We literally don't know because we don't know what we're doing. Is it a sparse coding phenomenon? In which case, 1,000 electrodes may not be better than 10,000 or may not be worse. Now, the analogy I use is right now we are trying to play Chopin on the piano, and we're banging the entire keyboard with our forearms. That's our therapy.
What's the way out of this? I don't know. We've been banging on the piano for 20, 30, 40 years. You're saying that it hasn't gotten better necessarily. The stimulation SCS has kind of remained the same. So is it going to get better?
It's about to get better, though. I'll bet you most of the stuff is about to get better because we have better tools. We didn't have optogenetics. We didn't have optical reporters that allow us to visualize with genetic specificity what we're activating. We haven't been using them in combination as well to use the clinically viable, to understand the clinically viable eSTEM stuff. We haven't been creating structures, but you're starting to see that through S.P.A.R.C and the BRAIN Initiative to say, let's connect the small animal to the large animal data to the humans and have people talk and say, what do you see here? When does it matter in large animals and humans? And when is it not predictive? How do we start to identify what variability in anatomy matters? Those structures are starting to be put in place. And you're seeing things like Saluda doing ECAPs in the home with closed loop systems to keep A-beta activation for spinal cord stimulation and pain constant and things like that. So we're starting to get these tools that always start off in animals first are starting to become these closed loop development. But it's all about creating structures for parallel translation. I think if clinical researchers are doing their stuff without really smart people with really sophisticated tools in the animal field, they're doing themselves a disservice. I did that, quite frankly. We didn't know exactly what we were stimulating for effect, so we didn't optimize our therapy. But if animal researchers think they're going to do something in a rat without really understanding the process to get to clinic and working with those people and do something that's impactful, they're also out of their minds.
Yeah. Okay. So we have more tools. We have better methods to be able to tease out the effects and everything like this. So
On
maybe
systems for people to work together across small, large, and human research, as opposed to doing it in silos.
okay. Are we going back to your piano analogy? Are we going from using our forearms to play Chopin to using our fists? It's like better, but still far away.
That's better. I got interviewed by Colonel. I had discussions with Neuralink and stuff like that. And one of the questions that would come up with, if you had 10,000 electrodes, what would you do? And what pretty much everybody answered, I need them to figure it out. It's that chicken or the egg problem. I don't know until I have it. I don't know what it would be useful for, and I don't know if it'll be worth the cost. But most of us like to think we can figure out something, which is why most academic researchers go, yeah, that's great. Give me 10,000 electrodes so I can figure this out. That's fantastic. And a lot of industry people go, oh my God, that failure modes and effects analysis, all the points that can go wrong for trying to get something through to humans. In industry, everything's keep it simple, stupid. You want 10,000 electrodes to figure out what you need, but then you want the simplest configuration that's the least expensive you can for your supply chain and to make a business case. That's why Tim Dennison's doing some fantastic work to say, okay, how do we create scientific research platforms that aren't going to be the market approved device, but that can be used clinically to help define what's the most viable product for a business case. And you see a lot of people attempting to create these smarter platforms that have expanded functionality for early stage clinical trials, knowing it's only going to be used in limited hands that can be controlled, and then say, okay, what's the idiot-proof, simple, inexpensive one we can create so your grandma can use or your grandfather? I don't want to be safe.
Interesting. Yeah, that does seem learning from your mistakes and just being a little bit more humble going into it and just being like, well, we have no idea. And even afterwards, we're probably not going to have any idea, but at least we'll get closer.
No, we'll have a little better idea.
Yeah, exactly.
Throughout human history, we were doing leeches 150 years ago. We evolved from chimps 5 million years ago. It's amazing to me that there's a set of people who always think, yeah, I understand the human history. We were pretty much always wrong and there were paradigm shifts every 50 years, but now I'm pretty sure we're done. It just amazes me. It's like there's that famous person who said, we don't need patents because everything that was ever used, everything that's ever been, that would be useful has been patented. So no, we don't know. We are ants trying to figure out how to carve.
I think that was like a patent officer in the 1890s and think of all the stuff that happened since then. But it really is good to acknowledge your limitations and how small of a part you are, and you're standing on shoulders of giants, and people stand on your shoulders as well.
Well, and plan for it in what you do. We should be looking. So one of the things that I wanted to mention to you is we know this from drugs. So we see a lot of these ends of 30 studies that people are saying we just cured something or blah 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 in our field. That's essentially a phase 1 study in drugs, and it used to be under 16% of phase 1 studies went on to successful phase 3 studies. We know the majority of this doesn't scale. It's like polling 30 of your neighbors for the presidential election and thinking you're going to be able to predict the election. Work. No matter how you randomize, you're still going to get nuisance variables and things like that that you didn't predict. It's why there's the Oxford levels of clinical evidence, and really the true class 1 evidence is many RCTs doing the same sort of therapy in a very similar sort of way and them all showing it works. That's our class 1 evidence. Right now, in a lot of these things, like if you look at AVNS, there's people doing it 100 different ways, and it's hard to even aggregate the data. And they're doing things like stimulating with literally the exact same current and pulse widths that we do for invasive cervical vagus stimulation because, well, it worked for an invasive implant on the trunk, as opposed to these are very different nerves and a pathway, different coupling. And that's always crazy to me. People are taking something that's completely not analogous and porting and expecting it to be the optimized configuration. Just doesn't make sense to me.
Yeah, I don't know. It seems arbitrary. It seems random. Like what you were saying, the small percentage make it through the phase 1. And I wonder how many like really good therapies were out there just because of chance. Somebody had a bad day somewhere at some point that fell through.
I don't think the stats work like that though, man. I don't think it's like somebody had a bad day somewhere. It typically, if you look in the history of all the targets that have been used for epilepsy within the brain, it always starts off with an N of 8 study by some neurosurgeon where everything worked and it got interest. And then they did an N of 40 study and then it didn't work. And then they tried to figure out why the N of 8 worked and the N of 40 didn't. Was it a fluke of the stats and things like that? Brain stimulation for epilepsy was done, or serious trials were done like in the 80s. And we just had the Sante approval and the NeuroPace approval in the last 5 to 10 years. So low end studies aren't necessarily predictive of, will this work in 1,000?
Yeah. I guess that's what I was saying is more like the randomness and having a bad day and like more, I don't know, being included or excluded from a study and that kind of stuff. But
well, that becomes more of a problem with these smaller ends as well. But the problem with that is, again, you're supposed to make it good enough where if you do a 100 patients and it doesn't work because one person had a bad day, then guess what? It's not that good of a therapy. It's not strong and reliable enough, especially for an invasive implant. We, by definition, because we're doing smaller ends and in many cases more invasive stuff, we actually have to have a really big effect in blood pressure. You get an orally administered drug that changes your blood pressure 8 millimeters of mercury, you're going to get FDA approved. You're really going to need, for an invasive implant, you're going to need 20 millimeters of mercury or more just because of the risks associated with an invasive implant. Plus the fact that you got to maintain it for 5 to 10. A lot of things we don't consider is, what if the startup company fails? Now I got a piece of hardware in my neck and I've got nobody who knows how to maintain it.
A DeLorean that you're like, I can't get the parts for this.
And it's not great to have it in there if it wasn't working, but what if you were one of those responders and it was making a huge life difference? But for me to get this implanted, I'm taking the risk that the company's going to be there in 5 years. Whereas with drugs, you can stop taking a drug.
Yeah, that is a really good point. Yeah, there's a lot of issues with this. Yeah, biology is very noisy versus like engineering or science or something like this. You might get a 3% variance there, but in biology, you'll get a 30% variance. And so like you said, the effect has to be huge. And then again, to make up for all the downsides. The
other thing you have to realize is the other disadvantage of small ends is when you do get a statistically significant effect, you're often overestimating its mean. And the reason being is because for it, say you have a modest effect, for it to be seen above the noise, you have to get lucky with the noise. And it has to bring your modest effect into the range where you can detect it, which means it's giving you an artificial feeling for the intensity of the effect, the magnitude of the effect. So you got a lot of things that are, say, under your noise floor, but have a little bit of an effect, but you can only see them when noise takes it well above it. And so it's not nearly as a big effect as you see. That's why in a lot of these studies, when we follow on and do more studies, we go, okay, it doesn't work nearly as well as that, because it was tip of the iceberg problem. It was just the little bit of ice that was right above the water.
So we have to redo statistics. Is that what you're saying?
Yeah, I think the problem we have is statistics aren't even definitive. A lot of this stuff is evolving. We don't even know how to do it. You've seen that with hypoglossal nerve stimulation for sleep apnea. You talk to people at Apnex, S.P.A.R.C, or Thera, which are very similar in terms of technologies, but one passed a clinical trial and two didn't. They're actually all going to say it was clinical trial design and being able to account for them. And some randomness as to what the FDA allowed in one study versus.
Crazy stuff. I love talking to you. I think this is a really eye-opening kind of thing. But this episode's been going longer than most. But as we're finishing up, is there anything that we didn't talk about that you wanted to mention?
Yeah, I think it's an exciting time for the field. I don't think it's a nihilistic thing. I think we're at the point where we can get these technologies small enough and minimally invasive enough where we can do things with the nervous system with lower risk profiles. But the problem being, lower risk profiles for the implant. If we don't know the mechanism, though, then the possibility of us changing the system in a bad way 5 years later becomes realistic. And so if we want to get to where we want to get to, we really do need to have our mechanistic understanding parallel our technological ability to put things on small chips.
Yeah, definitely. Like I said, I was always more practical. If it works, we don't need to know all the details. But no, you make a very good case, very good point on that. Thank you so much for coming on and second time and stirring the pot a little bit and being a little bit more antagonistic. I love it.
Yeah, that's right. You got problems, come to me. Bring it on.
We'll make them bigger problems. Thank you so much for coming on.
Always great talking to you.
Hope you enjoyed the show and were able to learn something new, bringing together different fields in novel ways. Until next time on the Neural Implant Podcast.